Thank you so much for the invitation. I was asked, let me project this. Can you see the right screen?
Yes, that's perfect.
Yes, okay. Yeah, I was asked to talk about current status of Treg therapy in transplantation. I mostly focus my talk on the clinical aspect of Treg cell therapy. Okay. So I have some disclosures and I have some financial interest in this space, and then these are companies I advise and co-funded. So Treg cell therapy in humans started with the first case report in 2009 and out of Poland. And that was in 2 cases, one chronic and one acute GVHD. And ever since then, until now, there are about 50 clinical trials of Treg cell therapy, mostly in North America and in Europe. There's some Asian countries also have conducted Treg cell therapy. As you can see, we at UCSF have now conducted conducting 10 clinical trials of Treg therapy. They're all in early phases, phase 1 and then phase 2A therapy. So I'm going to mostly focus on our own effort to give you a flavor on how it is like. I think it's quite representative of what's happening around the world. And so this is UCSF Treg cell therapy portfolio. And of the 10 clinical trials we have conducted, they are in autoimmune diseases and type 1 diabetes, lupus, and pemphigus, and also solid organ transplantations in kidney and liver, and islet transplantation. We just recently started a clinical trial of allogeneic Treg cell therapy for COVID-19 associated ARDS, and have infused 2 patients so far. It's too early to report on that. And then we're also planning CRISPR-edited Treg cell therapy. That's we hope to launch in the next 2 years or so. So our program has been in existence for about 10 years. So all Treg cell therapy at the start need to think about how to produce the Tregs. And then generally all the groups and follow similar approach. You start with peripheral blood, and then either leukapheresis or whole blood, and purify Tregs. And they're generally max purification and fax purification. We use fax purification so that we can use 3 colors, CD4, CD25, and CD127 for high pure isolation of the Tregs. And because we expand these cells for a long time without rapamycin, and then this high purity is really important to prevent contamination, outgrowth of contaminating cells. We have 2 general protocols of growing Tregs. The first protocol, we call it Gen 1, is polyclonal expansion using anti-CD3, anti-CD28 beads in the presence of IL-2. And we give 2 rounds of stimulation and harvest in 2 weeks. But others have also used similar approach and then can expand the cells for a month before harvest and then use for infusion. Our Gen 2 Treg therapy is specifically designed for transplantation indications. We use donor antigen to stimulate the Tregs to proliferate in the first round of expansion. And then the donor antigen we use are B cells that have been previously activated using CD40 ligand. And then these activated B cells are very, very potent at driving Treg proliferation in the presence of IL-2. By 7 days after stimulation, all the cells that remained in the culture are donor specific. And then we give them another round of stimulation to expand their number and before harvest and use for, and perform release assays before infusion. And let me first tell you about this large international effort of cell therapy to promote tolerance in kidney transplantation. This is cell therapy, not just Treg cell therapy. This particular study is called a one study, have multiple projects within it. And then they involve 7 sites listed, mostly Europe, and then 2 in the US, in Boston and in San Francisco. And then these 7 groups have different cellular products. In Regensburg, and they use the regulatory macrophages. At NANS, and they used these tolerogenic DCs that's derived from the recipient. These MREGs are derived from the donor. And then there are 2 sites, Berlin and then UK, use polyclonal Tregs. And then 2 sites in the US use donor alloantigen reactive Tregs. We call them DAR Tregs. Our Tregs, as I mentioned, is stimulated using B cells, whereas the Boston Treg are stimulated using co-stimulatory blockade, and then combined with donor antigen exposure. So here is the Treg subgroup, and then the type of Treg they infuse, and then dose of Tregs infused. So in total, they are 60 patients enrolled in the cell therapy group. 38 of them were treated, and then all 38 patients completed their trial. And this is a phase 1 study to assess the safety of cell therapy in kidney transplantation. And then biopsy proven acute rejection is the primary endpoint. Right, here's the data summarizing the biopsy, the primary endpoint. And then this is the intent to treat analysis and per protocol analysis, comparing the cell therapy group. This is a collection of all the cell therapy, MREGs and DCs and Tregs. And then with the reference group that's also conducted under the umbrella of the one study at the same time. So you can see there's no difference between the reference group and the cell therapy group in terms of a biopsy proven rejection by either intent to treat or per protocol. So cell therapy used in this context is quite safe. So in terms of other safety parameters, and then the study comprehensively assessed adverse events. And in general, there's no increase of adverse event associated with cell therapy group, these blue bars. And what's interesting, actually, there's one noticeable drastic difference between the 2 groups, is that the cell therapy group have much lower incidence of infections and infestations. And if you zoom in on this, the definition of infections and infestations, and these are mostly viral infections, and that are greatly reduced in the cell therapy group. This is unexpected and a surprising finding. It shows that indeed, by giving a immunosuppressive regulatory T cells, we found that the therapy is quite safe and then actually did not interfere with antiviral immunity. So that's all very good news. And the next step in this one study consortium, the one study consortium completed the study, but a follow-up, actually the UK group launched a 2 study aimed at assessing efficacy of this approach. And so everybody is interested in efficacy. We are as well. When we're thinking about designing a trial for efficacy, we first thought about what organs to use. In fact, the immune system responds to different transplanted organs differently, with liver being the most tolerogenic organ, whereas the bone marrow and the intestine and the lungs are less tolerogenic. In fact, in the mouse model, liver transplantation is spontaneously tolerant. So you can give an allogenic liver, and then the mouse will not reject the liver and then can live with that liver. And not only that, after liver transplantation, and you can actually put on a skin transplantation from the same donor, it will not be rejected. It's really very robust immunological tolerance to the graft. And then when we analyzed the mechanism of tolerance, of the spontaneous form of tolerance, we noticed that, and here's a naive animal, you can measure donor-specific CD8 in this blue box and donor-specific CD4s. And then among the CD4s, you can see there's some of them are Tregs. But if the mouse has been previously transplanted with a allogeneic liver, if you measure the donor-specific CD8, they're mostly deleted. And then the CD4s are not completely deleted, but they're greatly enriched for regulatory T cells. So we thought, well, this is a really good rationale for testing whether by giving more regulatory T cells to humans who don't have spontaneous tolerance to liver transplantation, we could improve the chance of having liver transplantation tolerance in humans. So in humans, liver transplantation tolerance don't spontaneously develop like in the mouse models, but they can develop over time. And if you take patients who have been transplanted for 10, 20 years, the chance of having tolerance can be as high as 50 to 80%. And then this is a graph summarizing patients who are within 6 years after liver transplantation. And then they undergo medical supervised immunosuppression withdrawal. So the x-axis is a reduction, percent reduction of immunosuppression from the time of the enrollment. And then the y-axis is their rejection-free survival. As you can see, most people at this early time after transplantation can actually tolerate up to almost 50% reduction of immunosuppression. And after that, then they start to have rejection episode. And ultimately, about 13%, 15% of the patients can are indeed tolerant to the graft. They can come off immunosuppression completely. And what's more is that when the patient have rejection episode, you can treat the rejection by escalating immunosuppression. And then that doesn't have long-term consequence to the graft or the patient. So we think this is the perfect scenario to design an efficacy clinical trial to assess Treg infusion in promoting liver transplantation tolerance. As you can see in this graph, there's a lot of room for improvement. So we designed this study enrolling adult liver transplant recipient. They have to have a living donor because we need their B cells to manufacture donor-reactive Tregs. We enroll them 2 to 6 years after transplant. We performed the power calculation based on this prior data that we need 9 to 11 patients. If there's a strong signal of tolerance activity by the Tregs, we should be able to detect it. Our target dose was 300 million. And then if the patient receives 300 million of donor alloantigen reactive Tregs, they're permitted to withdraw immunosuppression 100%. We also define a minimal infusible dose. This is because we feel, we estimated below 100 million will not be sufficient to make an impact. So for a patient who do not have enough cells, 100 million cells to infuse, and they are withdrawn from the trial. But if they have between 100 million and 300 million Tregs for infusion, they are allowed to withdraw immunosuppression up to the 75% point, which is our primary endpoint. And the study enrolled a patient at UCSF, Mayo Clinic, and Northwestern. And then we also decided that the timing of the Treg infusion is right before the scheduled 50% reduction of immunosuppression, so that we can see if this curve can be flattened out by this treatment. So we have completed this study, and this gives you a sense of the magnitude of the study, trying to enroll 11 patients into the study. And then in total, 322 patients were screened. Many of them were not eligible because of medical reasons. And then there are also patients not eligible for non-medical reasons, and this tend to be location and convenience and the ability to participate in clinical trial. And then they are among these, and then 25 were eligible by chart review, and then some of them, 10 of them, declined to participate in the trial. So among the 15 consented, 5 did not meet the study criteria upon further examination. So 10 were enrolled into the study. One of the subjects actually experienced the rejection immediately after drug withdrawal. So he was dropped out of the trial before the initiation of Treg manufacturing. We manufactured a Treg for 9 subjects. The big surprise to us was that 4 of the subjects, we could not manufacture 100 million doses. So we end up infusing only 5 subjects. And then here is a summary of the 5 subjects who received Treg therapy. 2 of them received the target dose above 300 million. The target dose is 300 to 500 million. And 3 of them received the suboptimal dose between 100 million and 300 million. And during the manufacturing, we labeled the Treg with deuterium. This is a stable isotope so that we can track the dynamics of the infused Tregs using deuterium by measuring deuterium in the DNA. And this allows us to assess the engraftment of the Tregs and the persistence of the Tregs. Here, each line is 1 subject, and then their subject ID and then dose of the Tregs infused is summarized below. So you can see the more Treg that was infused, the higher the deuterium level we detected. In fact, if you calculate the area under the curve here, and then you plot it against the dose infused, and we see a direct straight line correlation. This really shows that the detection technology is very robust, and also that at even the highest dose we infused, most likely we did not hit the engraftment ceiling. So the patient can actually take more Tregs, but the challenge was really that we could not manufacture Tregs for these subjects. So why is that? This is completely unexpected. And then at the same time, when we were conducting this ARTEMIS trial, we also manufactured donor-reactive Tregs for other trials. When we compared the outcome of Treg manufacturing for ARTEMIS versus non-ARTEMIS subjects, we saw statistically significant differences in terms of the final product yield. And then this is in part because we isolated less Tregs on day 0 after from the peripheral blood, and all the orange dots are ARTEMIS. So there's also a strong correlation of how many Tregs we put into the culture versus how many Tregs we are able to collect at the end of the expansion. And so part of the reason is because these patients have less Tregs to begin with. Another reason for this is that Tregs from ARTEMIS patients don't expand very well. Here is a Treg product yield as a function of expansion over the 16 culture manufacturing period. As you can see, most of them have lower fold expansion compared to the non-ARTEMIS subjects. As I mentioned in the slide for the mouse study, that liver can delete donor-specific T cells. And in the mouse, this was clearly selective for CD8 cells. But in humans, we wondered whether donor-specific Tregs were deleted or some other way, by some other way dysfunctional. So we directly measured donor-reactive Tregs using the CTV dye dilution assay. And then for this analysis, we actually collected sample from another cohort of prospectively collected from pre-transplant 6 months and 2 years after transplantation, and then measure their donor stimulated T cell proliferation, Treg proliferation versus a mixture of alloantigen stimulated proliferation. So you can see here, with donor stimulation, you see diminution of response over time after transplantation was with the pan-allo, the mixture allo reactivity, and then that was not changed. And here's a summary of all the patient data from pre-transplant to 2 years after transplant was donor stimulation versus pan-allo stimulation. Here's a pairwise comparison, and then showing quite consistent loss of donor reactivity. We think this loss could be dysfunction, as Marisa mentioned in her talk, but for her it's T effector cells, and for us here we're talking about Tregs. It could be also deletion of these cells by the transplanted liver. So this is, as you can tell, it's quite disappointing. We want to see if we have a chance to detect efficacy, but in the end, we were not able to infuse enough patients to be able to assess efficacy of this approach. So where to go from here? I think this is a published study published in 2016 by a Japanese group in liver transplantation, trying to promote liver transplantation tolerance using regulatory T-cell therapy. I think this gave us a ray of hope, and then also thinking about how to redesign our study to better assess whether Treg therapy have a chance of inducing tolerance. So in this Japanese protocol, they infused regulatory T cells very early during liver transplantation. Before Treg infusion, the patient also received the cyclophosphamide as a lymphodepleting agent. So if you think about Tregs as a way to balance out the effector cells, deleting the effector cells is a good approach to synergize with regulatory T cell infusion. What's really amazing that they followed this protocol of cyclophosphamide Treg infusion and drug withdrawal, and it was able to observe 7 out of the 10 patients achieving tolerance. And then this is published now 5 years ago, and I think these patients are now approaching 10 years after transplantation are still remaining tolerant, as far as I know. Another approach to this is when patient lost donor reactivity, we may be able to engineer donor reactivity using chimeric antigen receptors. In fact, the use of chimeric antigen receptor engineered Tregs have been reported since 2005. Around 2016 and 17, there are 3 papers reporting the development of anti-HLA-A2, anti-alloantigen chimeric antigen receptors for transplantation indications. So in the case of ARTEMIS patients, perhaps we can give an anti-HLA-A2 CAR to restore their alloantigen reactivity. And in fact, we are also able to use anti-HLA-A2 to redirect Tregs. Here we use a knock-in approach. We put the CAR into the TCR locus and then delete the TCR at the same time. As you can see, after editing, most of the cells express a CAR but lost the CD3 expression. And then they respond to HLA-A2 by upregulation of activation antigen. They traffic to the site of the A2 positive islet transplantation in the NSG mice. And then they function to reduce the lethality of xenogenic GVHD in the NSG mouse model. So these are approaches we and others in the field are developing, and then hoping to continue to push forward for an efficacy trial of Treg cell therapy in transplantation. So in summary, we have about 12 years experience of Treg therapy in the clinic. There are more than 50 trials and more than 100 patients infused. Up to 7 billion Tregs have been infused per patient in single dose. And then autologous Treg manufacturing is feasible, but it's not easy. Treg therapy have been well tolerated in patients. And then in some studies and favorable changes, immunological biomarkers have been observed. And then there are lots to do. We need more robust cell manufacturing technologies. And then we need that approach to generate antigen-specific Tregs. And as a field in general, we have not seen efficacy yet. We don't know which disease will see efficacy first, what antigen to target, and what dose to use yet. And then tolerance most likely will need Treg-friendly adjunct therapy, but this yet need to be defined. So with that, I'm going to conclude by thanking my many long-term collaborators in the clinic and also in the GMP manufacturing arena, and also my lab and funding bodies. Thank you very much.